Hello, and welcome to today's session on localizing sounds. In recent sessions, we've talked a little bit about the physical stimulation that gives rise to our psychological experiences of hearing. We talked about the phase, amplitude, and frequency of the auditory stimulus. We've also talked a little bit about the physiology that's associated with our hearing sense. Today we're going to specialize on how it is that we localize sounds. So let's jump right in. When we perceive a sound, we often simultaneously perceive the location of the sound. We don't typically have to work for that. It seems to be information that's given to us right off the bat. Once we can identify a sound has occurred, we almost always know where it's coming from. And as I mentioned in a prior lecture, when we're born, assuming that we've had anywhere near the typical 9-month or so gestational period, we can localize sounds even as soon as we're born. So you can snap on one side of the baby's head and the baby will orient that way, and on the other side the baby will orient that way. And this is true for lots of different species. So it appears to be the case that our brain is wiring itself to localize sounds even when we're developing in the womb. It's a very fundamental capacity that we have. And we can pick this information up even though it's the case that there's nothing in the sound itself that tells us about its location. So it's really quite interesting that a given sound contains absolutely no physical property that designates its location. Again, earlier we talked in a prior lecture about a sound's phase or its frequency or its intensity or amplitude. We talked about all those properties, but there's nothing about those properties that intrinsically conveys the location of the sound. So what we can say then is the ability to localize a sound must be caused entirely by neural events, since we can't pick up positional cues from the stimulus itself. So in today's discussion, we'll be trying to understand some of the different tricks that our nervous system uses to help us localize sounds. Ideally, it would be adaptive for an organism to localize sounds in all three spatial dimensions. So we can think about localizing sounds in the z-plane, and also in the y-plane, and in the x-plane. That would be ideal if we could do all three of those really, really well. For simplicity today, we're going to reduce the dimensionality across which we will investigate this phenomena of localizing sounds, and we will omit the depth dimension, the z-axis, but I would like to return to that either later in this session or perhaps later in the semester. We'll come back to some of the cues that might be available to helping people localize the z-axis location of a given sound source. We're going to learn two other terms today as we focus on the two remaining dimensions in which we can localize sounds. One of the terms is pronounced azimuth, and this is the position along the horizontal plane or x-axis. The other one is more intuitive to us. It's elevation, position along the vertical plane or y-axis. And so just to help you orient on this, we'll be talking about localizing sounds in the azimuth, and I'll ask you to make this gesture with me. You can stick your finger out and go along like this, rotating across the x-axis, and we could go through 360 degrees, but here I'm just doing 180 degrees for simplicity. Okay? It also helps if we get the gesticulation down for elevation. Again, we're sticking the finger out, and then we can go up or all the way down. We can even go past the 180 degree mark further that way, although we don't typically need to do it that way. Okay? So, azimuth and elevation are the two dimensions along which we will localize sounds in today's discussion. And here's a diagram that just gives us some idea about that. Both azimuth and elevation are relative to head position, or more precisely at ear level. We can talk about how far we'd have to go over this way or over that way in the azimuth, or up or down in elevation. So we get the emergence of something like a sphere that would contain the points that we can specify by these two coordinates, the azimuth coordinate for the x-axis and the y-coordinate or elevation coordinate for the vertical expanse. Okay? So psychophysicists typically describe locations by using those two coordinates, one for the azimuth and one for the elevation, and we typically omit the z-plane, although we could come back and develop a more extensive coordinate system that would actually capture that as well. But that will be for a different conversation. We will not discuss that today. The two coordinates seem to be specified by the listener, or specified to the listener by different neural events. And we're going to try to understand what's going on in the neural response to these stimuli that might give us some clue about where the sounds are originating from, in the azimuth and also in the plane of elevation. Well, we'll first consider the neural events that pertain to the azimuth. It turns out that the cues here are a little better understood than they are in the elevation plane. They're also a little bit more complicated, interestingly enough. So we'll try to understand how it is that we localize so well in the azimuth. There's one other reason why we're going to spend most of our time today talking about localization in the azimuth, and that is that you and I are terrestrial animals. That is, even though we can go into the water, typically we spend most of our time on land. And so unlike birds, or unlike fish that are moving very frequently through 3 dimensions, and they need to move very precisely through 3 dimensions, typically you and I are moving just along the ground plane. And this renders the azimuth the most relevant plane for localization. You could make the argument that occasionally we might want to locate prey that are flying above us. Occasionally, in our ancient past, there could have been dangerous birds attacking us from different elevations, but for most humans, under most evolutionary conditions, the plane that really matters would be the x-plane, or the azimuth plane. So we'll spend most of our time talking about that. Okay, so we're going to talk about 2 primary cues that help us localize sounds in the azimuth, and the first of these cues would be interaural intensity differences. Interaural means between the ears, okay? Interaural intensity differences, and we frequently refer to these as IIDs. And we can describe the IIDs as the disparity between the amount of acoustic energy that reaches the left and right ears. IIDs vary with azimuth, the horizontal position, of the source, and the frequency of the stimulus, as we'll see in the next slide. So 2 factors are going to be affecting this particular kind of a cue. To where a sound is located in the azimuth, this IID. Okay, one is going to be frequency, and the other is going to be position. So let's see if we can understand this graph. It's a little bit busy. Let's see if we get the axes down first. On the ordinate, or y-axis, we have the interaural intensity difference expressed in dB. And we could make this dB SPL, or dB IL for intensity level, or sound pressure level, but we'll just leave it at dB for the moment. A 0 IID would correspond to no differences in the intensities reaching the 2 ears. So one of the ways to think about these kinds of recordings is to imagine that you have very, very small microphones that you could actually place just for a moment in each of your 2 ears. And we could then play various sound sources for you. We could play sound sources that are directly in front of you, or all the way behind you, or off to your right side, or off to your left side, or at any of the angles in between along this 360-degree expanse of the azimuth. We can play sounds, maybe at ear level, for any of those positions. And we're recording both from the left ear and from the right ear. And those microphones can register the dB level coming into your right ear and left ear for different stimuli located at different positions. So that's how we're making these recordings. And we're going to talk about the difference in intensity levels between the 2 ears, or the IID, interaural intensity difference. That'll be on this axis. On this axis, we have the direction of the sound source. So we can arbitrarily establish that 0 is going to be straight ahead of us, okay? And 180 degrees will be right behind us, and then we can pick a particular direction. We might say, for example, that will go 90 degrees off to this side, or instead 90 degrees off to that side. We typically take one of those at a time, and we'll talk about the 180 degree expanse, we'll say from here all the way over and around to the back, okay? 180 degrees. So across that range of different directions in which we might record binaurally these sources, we can ask what kinds of interaural intensity differences do we get as a function of this positional change, or the direction of the sound source. Here's what we see, something relatively systematic. For a 6,000 hertz tone, this is a moderately high tone. I would play one for you, and I think that you would be able to hear it live, but through this video, maybe not, because sometimes our recording equipment has a cutoff at maybe 6,000 hertz or even lower. So I'm not so sure it would come through, but we can play this in the classroom nonetheless. But for recording purposes, we were presenting a 6,000 hertz tone, and the acoustic energy differs by up to 20 dB in the 2 ears at the 90 degree difference. So I'm looking straight ahead at you at 0 degrees, 180 degrees is behind me. Off to this side, we'll say that we're off by now 90 degrees. You can see the right angle formed by my arms. So when we now have a sound source over on this side, okay, and we're recording the intensity coming to this ear and the intensity coming to that ear, when the sound that's being recorded is a 6,000 hertz tone. And what we see is that in this particular case, this is going to be my right ear, I realize it's on your left side, is receiving a sound that is actually 20 decibels more intense than is the sound coming to my left ear. So there's a 20 dB difference between the two ears, or a 20 decibel IID. If I now record not 90 degrees away, but we'll say maybe like 45 degrees away, or 50 degrees away, or a little bit more than 90, maybe something like 135 degrees, we can report what the IID would be in each of those cases for the 6,000 hertz tone. Now what's very interesting is we can play that same game one more time. Same arrangement, 0 is straight ahead, 180 is right behind me, off to my right, your left is going to be 90 degrees, but now we change the tone. Now we change the tone to just a 200 hertz tone, so this is relatively low. It's actually even lower than middle C. Now, this is something that would come across on the video if I were to play it. And what you notice is that the IIDs are really quite flat here. Okay? The interaural intensity differences are really quite flat. And to appreciate why we have this frequency dependence on the IID, let's just remind ourselves about a property that we mentioned 1 or 2 lectures ago. And that is this property of the relationship between the wavelength of a sound and its frequency. We've noted previously that these are inversely related. So if we have a relatively high frequency tone, we have a very small wavelength tone. And conversely, if we have a lower frequency tone, then we have a longer wavelength. So what is important is to think about how the size of your head might be playing a role in IIDs. So a given adult will have a head size of whatever it happens to be, and a very high frequency tone, like a 6,000 hertz tone, will have a very small wavelength. Imagine that the wavelength is coming in, okay, according to a longitudinal wave, and it hits this ear, and the wavelength might only be that big. Okay? If you can imagine that sound, unlike light, is capable of wrapping around different devices and different objects, the sound will attempt to wrap around. If I can anthropomorphize the sound, it'll attempt to wrap around. But the sound wave itself will be too small given the comparatively large size of the head. Let's say instead we had a different stimulus. It was a 200 hertz stimulus, and it has a much longer wavelength. Okay? Now, in comes that wave. It hits my right ear first and begins to wrap around, and actually will make it to the other side with almost no diminution in intensity. Okay? So the longer wavelength can wrap around a relatively large object like a head. So we can see that we have 6,000 hertz giving us really nice IIDs, but at the same intensity, a stimulus that now is at 200 hertz versus 6,000 hertz, a much longer wavelength, can wrap around our heads. This is interesting because this will be very contingent on head size. You can imagine a much smaller species, like, say, for example, a mouse, or a much larger species, like an elephant. Okay? The kinds of frequencies that are going to give you this type of difference will be very species specific. Even within a species, there will be some variability because adults have slightly different head sizes. If we look at newborn infants versus full-grown adults, the head sizes are going to be changing. So we get these IIDs that are going to be dependent always on some kind of frequency, and the particular frequency values will depend on the specifics of the situation, which member of a species, which species, and so forth. Okay? So we can see that we're getting nice IIDs for 6,000 hertz, but not for 200 hertz. Okay? So our IIDs are going to depend very much on frequencies. Okay, that was one of our 2 primary cues for locating sounds along the azimuth. Let's go to our second primary cue. It's also an interaural difference. So we still have an ID, but now we have an interaural time difference, an ITD, as opposed to an IID. Just to give you a heads up, in the literature on localizing sounds, you will sometimes see references to ITDs. Equivalently, some researchers prefer to refer to ITDs as interaural phase differences, but they mean the same thing. That would be an IPD. And just to help you understand that, first let's understand what an ITD is, the disparity between the time of arrival at the left and right ears. Okay? So now I'll take a sound that's coming toward my left ear. I know this is on your right side. Okay? When the sound is coming from that location, it'll reach this ear first, it'll reach this ear second. So there's going to be a subtle time of arrival difference between the 2 ears, and that timing difference might be something that the neurons in your auditory pathway could begin to compute and use to help you localize a sound. As you may recall, When we talk about the phase of a stimulus, we're talking about its relative position, either in space or in time. So when we have a timing difference, you could convert that into a phase difference. There are really two different ways of talking about the same phenomenon. So some researchers prefer to call interaural time differences interaural phase differences. They mean the same thing, those two phrases. So ITDs vary with the azimuth along the horizontal plane, and here's an example of that. So here we have an interaural time difference on this axis. So we have a new y-axis. It's a different unit of measure. This is now expressed in a time-based measure, milliseconds. I do want to point out the specific unit. This is milliseconds, and look at the range over which these are operating. This is 1/10 of a millisecond. This is 6/10 of a millisecond. So these are on the order of maybe a few hundred microseconds or millionths of 1 second. These are very brief intervals of time, even briefer than 1 millisecond. So it's interesting that our nervous system is that sensitive to these. As always, we have the same x-axis as we did the last time around. That is to say, we have 0 straight ahead, we have 180 degrees behind us, and then I'll go off to my right one more time, 90 degrees on this side. Okay? And so if we take that example one more time, if the sound source is coming from my right, and it's clicking away, and I've got my recording devices in each ear, just inside of each pinna, I can now have the sound come and hit my right ear first, my left ear second, and we will get a very subtle difference in time of arrival. Again, something like 0.6 milliseconds, which I think translates to 600 microseconds. But then if we go to slightly different angles, we're going to get slightly different ITDs. Okay? So there's a relatively reliable relationship between the position of the sound source and this interaural difference, in this particular case, an interaural difference that is measurable in time. Okay, so ITDs are largest, in this case 0.6 milliseconds, when the sound source originates from directly to my right or directly to my left. Okay. So we've talked about IIDs and ITDs, interaural time differences, and there's a well-known theory of localization which is called the Duplex Theory of sound localization, which relies very heavily on both of these cues. ITDs, interaural time differences, are used to localize low-frequency sounds, and IIDs are used to localize relatively high-frequency sounds. So there's nothing about using IIDs that would exclude you from using ITDs, and vice versa. These can be used in combinations, and maybe they're more successfully used in different ranges of frequencies, as is suggested here by this theory. Some interesting holes or lapses in our capacity to judge localization. Localization for adult humans is particularly poor between 2,000 and 4,000 hertz. We're not equally good at localizing all frequencies. Some of the frequencies are very well localized, and we can localize them to maybe 3 or 4 degrees of precision. Others we localize less precisely. We might need something like 20 degrees difference before we can localize those sounds. So this particular range is a range in which we're pretty poor at localizing sounds in the azimuth plane. Okay? ITDs and IIDs are both vulnerable to certain ambiguities that we'll see in just a moment. Specifically, a given ITD or a given IID, interaural intensity difference, can arise from more than one location in space, as we'll see here. So this graph is similar to what we saw a moment ago, but we need to pull out some new information. So let's see how we do that. Here we're talking about interaural intensity differences, so we're back to decibels on this y-axis, and now it's always, we have the direction of the sound source. Okay? Just so we get a little diversity in how we're thinking about this, I'm going to ask you to excuse me as I turn my back to you. I'll get into your orientation. 0 degrees will be straight ahead, 180 degrees will be behind us, 90 degrees will be off to this side. Okay? So what we can say is that when we're talking about interaural intensity differences, and let's say we have a relatively high pitch, a high frequency of 6,000 hertz, we talked about how if that's coming from this side, that might not be able to wrap around my head, and I'll have greater intensity over here than I will over here. But what's interesting is you can imagine that if I went to not 90 degrees, but say 45 degrees versus 135 degrees, each of those is displaced by 45 degrees from the 90 degree angle. Okay? So 135 back here, 45 degrees back here. Each of those will create very comparable IIDs. So I will have some notion that the sound might be coming to me not from straight ahead, not from directly behind me, and not from off to this side, but there is a huge ambiguity about whether it's coming from 45 degrees away from due right, if you'll allow me to use that phrase, or 45 degrees this way from due right. I have actually the same interaural intensity difference in those two cases, so there's a moment of confusion here. Here I'm picking 2 points somewhat arbitrarily. You could pick other points. You can see that we're getting comparable levels of IID from those two very different directions. Okay? So we would have no basis for distinguishing those two. And this is not true, again, as we said before, for the 200 hertz tone, because this lower frequency tone is going to generate virtually no IIDs for us. Okay? The same kind of ambiguity, though, that we referred to for IIDs, you'll see in a moment, will pertain to ITDs. There is one more point, though, to make about this. We get the same IIDs for the two horizontal positions, front versus back. And so if you are an experimental psychologist, and you're engaging in these localization kinds of behaviors, or kinds of experiments, you'll notice a very interesting aspect of behavior. And that is that people will frequently make front-back reversal errors. That is, they will almost never tell you that a sound coming from this side is coming from that side. They will almost never tell you that a sound coming from here is coming from here, but they will make a 180-degree error confusing the front from the back. Now you might say, how can they do that? It seems like front and back are very, very different from each other. They're 180 degrees apart. They're not just off by 1 or 2 degrees or 7 or 8 degrees. They're off by 180 degrees. Why would that be the case? Well, when we think about how the nervous system uses the cues available to it, maybe it does make sense that we would have front-back errors. So over here and over here, when we're separated by 180 degrees, we have actually no interaural intensity differences. When a sound is coming from straight ahead of me, my two ears receive that sound at the same time. If it's coming from straight behind me, my two ears receive the sound at the same time. I have a zero IID in either of those cases. So with respect to interaural intensity differences, I have basically no reason to believe that something's coming from the front versus the back. As we'll see toward the end of this discussion, we actually use some other kind of a cue, not an interaural cue, to help us distinguish front from back, but that's an ongoing problem that our species has. We're not really good at localizing front versus back, very intriguingly. Okay, that was the story for IIDs. Let's go on to ITDs, interaural time differences, as we mentioned, equivalently interaural phase differences. So now we're back to this axis, which is a time-based axis, and it's measured in milliseconds, actually fractions of a millisecond. And as always, we have 0 to 180 degrees here, and the same kind of issue arises now in time. I'll go back to my earlier orientation, if you'll excuse my back one more time. Back there is 180 degrees. In front of me is 0 degrees. Off here is 90 degrees. Okay? And then I might go, say, 10 degrees this way or 10 degrees that way from 90. That'll put me at 80. Or 100 degrees, and in each of these cases, the sound is going to hit my right ear first before my left ear, okay? But the time of arrival will be comparable at 80 degrees versus, say, 100 degrees. So we can find 2 points that are going to generate exactly the same ITD, and we would have some ambiguity there. And again, as always, this will be true for the time of arrival, front versus back. The sound coming from straight ahead of us and straight behind us will arrive at the two ears at the same time. We don't get an ITD in that case. Okay, so really interesting to think about this. It's also interesting to think about this as a possible metamer. I'll take just a small digression here because I think it's important to help you review for the final exam, which will be cumulative. You remember that we've talked about metamers. Metamers are physically different stimuli that are perceptually indistinguishable. So let's think about metamers in this context. We can talk about having a sound that is directly in front of us versus one that's directly behind us. Those positions of those sound sources couldn't be more different. They're physically different locations. Okay? And yet we have a lot of trouble distinguishing front from back auditorily. In most cases, we can't do that particularly well. So are these metamers, are these physically different stimuli perceptually indistinguishable, at least with respect to location? Here we can have a little bit of an argument. We could quibble about whether these are metamers in the same way that this yellow is a metamer. Remember, going back to earlier this semester, we talked about how we don't have any stimulation here, at the 590 nanometer wavelength. Instead, we might have, if you will, a green beam superimposed on a red beam. The red might have a wavelength of 650 nanometers. The green might have a wavelength of something like 550. And yet we're having the experience that's equivalent of a 590, even though there is no 590 present there. So we have very physically different stimuli that are creating the same perceptual experience. It's the failure of discrimination. In that case, though, the failure of discrimination is arising from some idiosyncrasy of the architecture of our visual system. Specifically, we get comparable levels of firing in the 550, 650 combination stimulus as we would in the 590 stimulus. So we have a failure of discrimination that arises from the architecture of the visual system. In the case that we have here, we're winding up with a failure of discrimination. We can't tell back from front. It seems like a gross error and possibly a metamer. But actually what's going on is not anything that's particularly unique about the structure of our auditory system. Instead, there really is no information available in respect to the ITDs or the IIDs that we had seen. That is, the time of arrival really is the same at the two ears. The intensity really is the same at the two ears. So the stimulus coming in is really quite impoverished. It's not that we're failing to discriminate because of some idiosyncrasy in our neural architecture. By contrast, in this case, we actually do have very different stimuli in the case of a 550 green and a 650 red landing to create the illusion of a yellow. Okay? That arises from some idiosyncrasy in our neural architecture. That's information that's available on our proximal stimulus is in principle dissociable, but over here we don't even get that kind of dissociable information on our nervous system. We have 0 IIDs, we have 0 ITDs, so it isn't too surprising that we fail on this task. An interesting way of thinking about metamers and to try to understand them a little bit more precisely. Okay, so localization errors, and we do make them, frequently front-back errors, but these can be reduced by moving one's head. Okay? However, head movements usually require a fairly long time. By neural standards, they might take 500 milliseconds or more. So head movements are only helpful in localizing sounds that are of relatively long duration. There might be a prolonged sound. There might be a sound that is short but repeated. And if it's repeated over time, and I can move my head from this orientation to that orientation, I can then bring the greater sensitivity of my localization processes that are going to be located just off to the right side of my head or just off to the left side of my head. Again, we're very poor straight on and straight back. So I can move my head this way or that way to bring in greater sensitivity and increase the interaural time differences or interaural intensity differences. But that will cost me 500 milliseconds, and in a jiffy that might be inappropriate. For other stimulus conditions, that might be all that I need to do. Right? So another factor that helps us minimize errors in localization has to do with the pinna, the outermost portion of our ears, and now we'll begin moving away from these interaural cues. Okay, let's just remind ourselves that sound bounces around the pinna before entering the auditory canal. I'm using bounces here in quotes. I'm being a little bit informal with that description, but it is true that sound sources do bounce off of various objects, and this is how echoes turn out to be apparent to us and so forth. So the shapes of our pinnas are going to play a very important role in the sound that's actually going to be reaching the tympanic membrane or the eardrum. The number and the direction of bounces depends on the direction from which the sound originates. So if the sound is coming from straight ahead and hits my pinna before it goes into my auditory canal, I'm going to get a slightly different acoustic signature than if the sound were off to this side or off to that side. So I get slight differences just based on the idiosyncrasy of the sound's position interacting with the unique shape of my pinnas versus, say, somebody else's pinnas. Okay? So this is equally true for vertical and horizontal displacements of sound. So unlike ITDs and IIDs, the pinnas could play a role in vertical localization. So most of today we've been talking about this azimuth and how we locate sounds along that dimension. Now we can also talk about the vertical dimension and recognize that the pinna could play a very helpful role here. I find this one fascinating. People can identify locations when they hear recordings from their own post-pinna auditory canal, but not from one's own or somebody else's auditory canal. Okay? So the notion would be that if I'm recording from my own auditory canal, and then you played that information back to me, I can localize really quite precisely, and I can say that a particular stimulus occurred in elevation location number 8, or number 7, or number 6, 5, 4. I have really good information about that. But then you play for me the recordings that were made from somebody else's pinna, and all of a sudden I can't do it. So it's almost as if my brain has learned through practice to locate sounds in the elevation plane by using my own specific pinna. If I have somebody else's pinna momentarily, by swapping out my recording for their recording, all of a sudden performance falls off. So here's a diagram of two similar but different pinna. You can see this person's pinna looks to be a little bit larger. It has slightly different convexities and concavities. Each of us has pinna that are a little bit different from everybody else's pinna. One other note about the pinna that I think you might find interesting. When we're born, our pinnas, like most of the other parts of our body, will begin to grow. Unlike the other parts of our body, our pinna never stop growing. And this is why it seems to be the case that when you look at elderly folks, they seem to have disproportionately large pinna. And that's because the pinna never stop growing. So an interesting fact about those. Okay, so here are two pinna from two different folks, and what we're going to show you here are some recordings that are made inside of the auditory canal from two different folks at various elevations. So we can have a sound source that's right at 0 elevation, or can have a negative elevation, lower than 0, or can have a positive elevation, and we can record from inside of my pinna or inside of your pinna. And the different pinna recordings are shown for one person in solid line and for a different person in dotted line. And you can see that for any given elevation, we're getting person-specific kinds of signatures across the frequency domain, because some of the ears, some of the pinnas, are going to absorb sounds of particular frequencies, others will reflect particular frequencies fairly well, and so forth. So each unique pinna produced unique waveforms in the auditory canal. And apparently we get used to these, and we can take advantage of particular kinds of signatures when we're localizing. So our brains adapt to sounds that come from our own pinnas, and we're less used to localizing sounds that would come from somebody else's pinna. Okay, so finally it seems that monaural cues, those are cues coming from just one ear, are sufficient for vertical localization via the pinna, since people can perform vertical localization equally well in monaural and binaural conditions. So just to make sure that's really clear to everybody, I could have recordings from both of my pinnas, and I now have sound sources that are varying along the vertical dimension. And my job as the participant in this study is to report that a particular sound came from elevation 8 or elevation 5. We can get into negative numbers if we wanted to refer to vertical positions that are lower than center, and so forth. And we can give a participant either a single ear's information or two ears. And it turns out in this particular case, most people are about as good with one ear as they are with two. So this would be a very clear difference between localization in this azimuth plane versus in the vertical plane. It looks like in the vertical plane, one ear would be just as good as two. Okay, so that's what we've got on localization. I hope that, as always, you'll make some notes as to what was particularly clear. And it's important to jot that down, and equally important to jot down a question or a comment about what was less clear to you, and bring those comments to class. See you in class.